Guys, welcome to my channel. In this series of videos, we are going to talk about the recombination process that occurs in prokaryotes. Okay, so what basically recombination is? So, recombination is a process of recombining genome or rearrangement of genetic material. Okay, um, let's take this. This is the DNA of one organism, and this is the DNA of another organism. Okay, now what the recombination means, these both are combined to form a new arrangement of DNA. This is called recombined DNA. Okay, it is usually occurs is in crossing over. And in case of bacteria, it occurs characterized by DNA transform from one organism called donor to another organism called recipient. Okay, so in bacteria, there are three types by which a bacteria causes transfer of its DNA to another bacteria. Okay, so these are the types. First one is conjugation, second one is transformation and transduction. These are the means by which a bacteria transfer its DNA to another bacteria for the process of recombination to occur. Okay, so now in this video we are primarily focused on this part, that is conjugation process. Okay, so conjugation. In conjugation, transfer of genetic material between bacteria through cell-to-cell contact. Okay, here cell-to-cell contact has to be made between two bacteria. This is bacteria 1 and this is bacteria 2. A cell-to-cell contact has to be made between two bacteria for recombination to occur. Here the transfer of genetic material from donor cell to the recipient cell takes place. Suppose this is called donor cell and this is called recipient cell. So the genetic material is transformed from donor cell to recipient cell. Now the donor cell donates mobile genetic material in the form of conjugative plasmid to the recipient cell. Okay, what do you mean by plasmid? A plasmid is a circular form of DNA present in prokaryotes. So this is the plasmid of donor bacterium. So what does it line means? Conjugative plasmid to the recipient cell that previously do not contain a similar element. So this recipient bacterium do not contain a similar plasmid like this. So this plasmid is transformed from the donor bacteria to the recipient bacteria to for the process of recombination to occur. Okay. Now the mechanism by which the process occurs, conjugation. So the prototype for conjugative plasmid is called F factor. F factor, or what is F plasmid? F stands for fertility factor. So what is F factor or F plasmid? Okay, let us consider this is the donor bacterium. Okay, and this is the mobile genetic material or conjugative plasmid. Okay, this is also called as F factor. Okay, the bacterium that has F factor is called F positive bacterium, that is male, and the other that lacks F factor is called F negative bacterium or female. Okay, or female. So the bacterium that has F positive factor is male or donor, and that lacks F factor is called F negative bacterium female or the recipient bacterium. Okay, so on the onset of conjugation, the F positive bacterium forms a sex pili which act as protoplasmic channel between and contacts recipient bacterium or F negative bacterium. What does it line means? On the onset of recombination, this donor bacterium forms what is called a sex pilis. Okay, this sex pilis is a means to connect the donor bacterium to recipient bacterium. Through these pillars, the genetic material of donor bacterium, that is F factor, is transferred to the recipient bacterium for the process of recombination to occur. Okay, the transfer of F element or F plasmid is processed by asymmetric DNA replication of rolling circle type. The F 5' end of the F element thought to be drawn into recipient cell where it copied in 5' to 3' direction. So what is it means? Here we have two strands of DNA, 5' to 3' and 3' to 3' to 5'. So when the process of recombination to occur, then these these two are joined by what? Hydrogen bonds. Okay, these bonds are break and these two strands become free. The 5' to 3' end, that is 5' to 3' end strand, moves to the recipient bacterium. The relaxes this is a protein that helps in transformation through sex pillars. Okay, this is important. Now what happens? The 5' to 3' moves here and the 3' to 5' stays here. So now what happens is in this bacterium, uh, the DNA replication occurs after recombination and the DNA replication occurs here, the recipient bacterium also after recombination. So here it in donor bacterium, the 3' to 5' strand is act as template, and in donor recipient bacterium, the 3' to, so this has to be recipient bacterium. Okay, um, the 5' to 3' act as a 5' to 3' act 3' stand act as a template for this recipient bacterium. Okay, so this is the uh, replication process. Now, meanwhile, the second strand of F element remains in the donor or male cell and serves as template for its own. So this is the 3' to 5'. This was the 3' to 5' end of the donor cell um, DNA strand that acts as a template for its. Thus, recombination is completed. So, thank you for watching. In next video, we are going to talk about the second way of recombination, that is transformation.